Hello everyone. Welcome to our presentation for CSE 115, Introduction to Biology and Chemistry for Computation. Today, our team will be diving into chromatography, a fascinating topic that connects both biology and chemistry. Before getting started, let me introduce my team. My team members are MST Sabrin Sultana Mim, ID 252-15073. MD Asif Rahman, ID 252-15204, Abu Bakr Siddiq Prottoy, ID 252-15292, Orgho Dev Shikdar, ID 252-15613, and myself, MD Arif Shahriar, ID 252-15470. Each of us contributed to different parts of this presentation. It was truly a team effort, and we are very much excited to share what we have learned and show you how this method plays a vital role in scientific analysis. In this presentation, we'll help you to understand the basic principles of chromatography, how it works, and why it's effective. Next, we'll explore different types of chromatography, such as paper, thin layer, column, liquid, and gas chromatography. We'll also demonstrate a simple chromatography experiment, which will give you the basic idea of it. And finally, we'll discuss some real world applications to give you a sense of how important this technique is across various scientific fields. By the end of this presentation, we hope that you will see chromatography not just as a lab procedure, but as a powerful tool in modern science. Now, Neem will talk about the introduction to chromatography. So, let's dive into the world of chromatography.
Thank you, Arif. Let me begin by giving you a basic idea about chromatography. Chromatography is a laboratory technique used for the separation of mixtures. It's specially used when the mixture contains substances that look similar but behave differently at a molecular level. The main purpose of chromatography is to separate individual components of mixtures so that we can study or analyze them. The method is extremely important not just in chemistry labs but also in scientific research, medicine, forensics, and even the food industry. Now let's look at how chromatography actually works. The process is based on what's called different migration. That means different substances in mixtures move at different speeds through a medium and allows them in separate. In chromatography, we have two phases, a stationary phase and a mobile phase. A stationary phase, it still the paper or plate, and a mobile phase, it moves like liquid or gas. When the mixtures is added, the different parts of move with the mobile phase. Some move faster, some slower depending on how well the dissolve or how strongly they stick to the stationary part because of the part separation from each other. Now I will talk about the different types of chromatography. These are paper chromatography, liquid chromatography, thin layer chromatography, column chromatography, and gas chromatography. That's all about the types. Now Asif will explain these use and importance. Thank you.
Thank you, Mim. Let's talk about paper chromatography. I will give a quick overview of paper chromatography. In this method, filter paper is stationary phase or solvent like water on alcohol is the mobile phase. As the solvent moves up, it separates components like ink, dyes, or plant pigments. We also calculate the RF value to help identify each substance. Now I will brief explain liquid chromatography. In this technique, the mobile phase is a liquid solvent and the stationary phase can be solid or liquid on solid support. It's known for high separation to ability. A common type of HPLC or high performance liquid chromatography which is widely used in pharmaceutical and environmental analysis. At least I will talk about thick layer chromatography or TLC. In TLC, the stationary phase is a thin layer of silica, aluminium, or glass or plastic. The mobile phase is a suitable solvent. The method is quick, simple, cost effective. It shows result by using UV lights or stain visualize the component. Now, Protoy will discuss about the other types of chromatography.
In column chromatography, we use a column packed with a solid stationary phase, like silica gel. Think of this as our filter. The mobile phase, a liquid or gas, then flows through this column, usually by gravity or pressure. This process effectively separates larger sample bodies. It's generally a slower method, but incredibly scalable, making it a stable in organic chemistry, labs, and for drug synthesis. Now, let's talk about gas chromatography. Gas chromatography takes the column principle, but uses a distinct mobile phase and insert gas like helium or nitrogen, which carries our sample. The stationary phase here is typically a liquid water inside the column. This method requires volatile samples, those that can easily turn into a gas. The beauty of gas chromatography is its high resolution and speed, allowing for incredibly precise separation. Because of this, it's widely used in forensics, in food analysis for quality and safety, and for crucial environmental testing. Finally, let's see how a gas chromatography system works in action, as shown in this diagram. It begins with carrier gas flowing from a cylinder through a regulator. We inject our sample into the injection part. The gas sweeps it into the column, which is kept hot inside an oven. Here the sample separates into its individual components. As each component exists, it's detected by the detectors, sending data to data analyzing monitor to show us clear results. Gas chromatography is truly an indispensable tool for analyzing complex mixture. With that, I will now pass it to our final team member, Orbo, to know about some real-world application of chromatography.
Thank you, Protoy. Now let's discuss about some real-world application of chromatography. Then I will demonstrate a simple yet fascinating experiment to show. Chromatography might sound complex, but once you start noticing it, you will find it everywhere, like in pharmaceuticals, forensic labs, food industries, educational and environmental science, biology labs, etc. In pharmaceuticals, it's used to test the impurity of drugs. A small impurity in medication can be dangerous, and chromatography helps detect and separate those tiny impurities. In forensic science, chromatography helps analyze toxins, drugs, or even ink from crime scenes. Yes, just like detective shows, but with actual science behind it. It's also widely used for food industries for checking if any additives or preservatives are over the limit and for ensuring overall food quality. Environmental scientists use chromatography to monitor pollutant in air, water, and soil. This helps identify contamination sources and keep economics safe. In biology labs, it helps the separation of plant pigment and even DNA compound, allowing scientists to study the molecule world more precisely. So as we can see, chromatography is not just for textbook. It's essential tool used across many fields that affect our everyday life. Now let's present a simple chromatography experiment that demonstrates how actually chromatography look like in real life. Hello everyone, today I'll conduct a paper chromatography experiment. For this, we need beaker with some water, we need some color pen or marker, and we need a piece of paper. First of all, I want to be brown. Then I will use the green color. Then I will use purple color. My coloring is done, as you can see. Now I'll insert this stick into the paper so that the paper don't fall on into this beaker completely. And I'll submerge this paper into water. Let's see what happens. As the time passes, water with those colors slowly travels up to the paper and spreading the color along the way. Each color moves at different rate, revealing the hidden pigment of the each single drop of color. Now the experiment is done. You can see how the color have separated on the paper. There are brown, there are blue, there are green, there are yellow, there are purple and pink color. The lighter colors move further and the heavier one stays closer to the bottom. This shows us that even different color can be made of different color. That's how paper chromatography works and help us understand what's inside a mixture. Thank you very much. To wrap up, chromatography is a powerful separation technique used across science, medicine, industry, and environment. There are many types of chromatography like gas chromatography, liquid chromatography, and what we just saw, paper chromatography. Each type is used for different mixture and substances. The simple experiment we just did today captured the core principle that different substances move at different rate in a medium, and that's the essence of chromatography. Using movement to reveal what hidden in the mixture. Thank you.